this week, researcher Riley was reading a research article authored by Daniel Soul and others about habitual desensitization. She learned that animals living in urban areas adjust their behaviors to suit their surroundings. As she stops to tie her shoe, she spots a squirrel, which sparks a research idea based on her earlier readings.
Hey Ari, yesterday when I was running, I saw this really cool squirrel.
Wow, no way. I always thought squirrels were really interesting.
Seriously? Me too.
Oh my gosh, we should study them. Hey guys, what's up?
Hey.
We just had a really good idea to study communication in squirrels.
Wow, no way.
Would you guys be interested in joining us?
Yeah.
The researchers begin to plan their experiment. They decide to study squirrel responses to predatory stimuli across the rural-urban gradient. And they're off to find the perfect locations. The researchers decided their urban location would be on Michigan State University's campus in West Circle near Beaumont Tower and Wells Hall. The rural location was set near the MSU farms on the outskirts of East Lansing. The researchers set up a speaker in discrete locations and did a playback experiment with a red-tailed hawk sound, car noise, and white noise as a negative control. They did 10 trials of each sound in each location and 5 for the control. The researchers have hypothesized that social environments will impact the mode of communication in fox squirrels. The researchers predict that if squirrels are in an urban environment, then they will rely more on visual communication. In a study done by Parton and his colleagues, they found that in urbanized environments, the eastern gray squirrel more heavily relied on visual alarm signals than auditory ones. Out of the 20 urban experimental trials, there were 2 vocalizations and 15 visual responses. Of the rural trials, there were 7 vocalizations and 13 visual responses. Most of the squirrel responses in both locations were visual, the predominant being tail flicks and tail flags as shown. We should see how communication is similar in humans.
Yeah. Let's get started.
The researchers hypothesized that a social environment will also play a key role in the communication of danger in humans. The researchers predict that if humans are stimulated by a startling noise in a solitary environment, then they will communicate with verbal responses. They set their location for the human trials in Holmes Hall of Michigan State's campus. They introduce a frightening stimuli of a book dropping to social and solitary settings and record the human responses. They also use a negative control of normal chatter to ensure the researchers were not eliciting the response. Out of 10 experimental social human trials, there were 5 visual responses, 2 auditory, and 2 that displayed both, and a single no response. Out of 10 experimental solitary human trials, there were 5 visual responses, 3 auditory, and 2 that displayed both. Most visualizations were defensive and fleeing mechanisms, and most of the auditory responses consisted of gasping. A study done by Arnell found that screams have evolved to be perceived as more fear-inducing, as well as increase the speed and accuracy for locating the source of the vocalization. This is extremely useful in solitary environments. This data shows the combined results of the human and squirrel experiments. It demonstrates that the predominant method of communication was visual over all gradients. After analyzing the fact that humans and squirrels responded in the same manner to frightening stimuli, researchers Ari and Ben wonder if the FOXP2 gene could be connected to this communicative behavior. According to the National Center for Biotechnology Information, the FOXP2 gene codes for a transcription factor which regulates the expression of a variety of genes. The gene is a requirement for proper development for speech and language regions of the brain. A squirrel sample was obtained, and the researchers are planning on testing the DNA to further understand the FOXP2 gene and whether squirrels have it encoded in their genome.